Of talks. So the first speaker is probability of evolution and the possibility of predicting evolutionary future.
Well, it's a great pleasure to be here, and I'm honored to be part of this inauguration of a new home for ICTS. And this is my opportunity to applaud for the broad vision of science that this institute stands for. The vision of the unity of science. And well, I'll be talking about biology. And of course, biology is not known as a hotbed of theory, but I will represent many of us who think that applying ideas of theoretical physics to biology is not only interesting, but maybe perhaps can even be somewhat useful. So, without further ado, I jump in and start talking about evolution. If I can figure out how to start the presentation. Okay. So very often when we think about evolution, we think about evolution in the context of natural history. The tree of life and the common origins of all of us, and plants and insects and frogs, and the process that took many billions of years to unfold. But evolution really can be also very fast, and we have a very different experience of it when we encounter almost every year the seasonal flu, right? And whereas my previous tree of life was an artistic rendition, this is a real genealogical tree reconstructed from sequenced genomes of different viruses that were isolated from the population over, as you see, 15 years. And what you observe here is, so the distances of these branches represent genetic distance, and what you see is this persistent drift of the virus away from its ancestors. And here you see that the population of viruses that in this slide was, I guess, sequenced in 2012, came to its common ancestor, so 2 or 3 years before, and then the population from, let's say, 2008, again, came to its common ancestor 3 years before. So this viral population turns over on a time scale of 2 or 3 years. And of course, the evolutionary process that drives it is the need for flu to mutate and escape human immune response. And our antibodies recognize particular sites on the proteins that are sticking out of a viral capsid. And then they bind to it and neutralize it. And as a result, the virus is under constant evolutionary pressure to innovate, to change these recognizable bits of protein. And this is done by accumulating mutations in its genome. And 5% of flu genome turns over in 10 years, which is really a lot. And here, this evolutionary tree of this flu virus is painted to identify particular, if you like, families with distinct viral epitope identity. And when people study the virus, the main emphasis is on trying to identify specific mutations that cause large changes in these recognizable bits of protein. So the question that I want to pose today is, can we understand this evolutionary dynamics well enough not only to understand what happened in the past, but also what's going to happen next year? So evolutionary dynamics is what connects this past to the future, and the key element of it unfolds in the viral population, as these new mutations, new genetic variants spread through the population. And it takes all of molecular biology and genetics to actually understand the, if you like, phenotypic, visible implications of a genetic change. And that is not even enough, because we also have to understand the ecology, which is to say the distribution of immunity in the human population, right, the hosts of the virus, in order to understand whether a particular change is going to be beneficial or adverse. So if you want to understand this sort of a priori, there is really a lot to understand. But we're going to take a drastic shortcut and put all of this science into a box and take a statistical genetics approach. We'll introduce a simple phenological model that will hide all this very important details and then try to achieve statistical prediction. So just a little primer on population genetics, which describes how alleles spread in population. So suppose this is a population at any given time, and all members of a population here are red, they're all genetically identical. Now this green mutation comes about, it changes so the shape of this viral protein makes it perhaps unrecognizable to immune system. That allows the virus to evade immune system, and as a result, this allele would be expected to spread in the population and take over. So after a year or two, all the viruses will inherit that particular mutation. But the process isn't really quite as simple as that, and evolutionary forces are not really deterministic, and most of the mutations that will come in, of course, first will occur in an individual virus, and their most likely fate is they're going to disappear. But mathematically, then you want to describe this in terms of a random branching process. So you basically say that a new mutation can so duplicate, or perhaps can have just single ancestor, or go extinct, all of that at random. And of course, this is very simple and has been understood a long time ago. This is what classical population genetics theory all worked out, how these individual alleles spread in the background of so identical population, clonal population. So the recent progress to which theoretical physicists contribute quite a bit was to actually understand what's going on in a case when there are many mutations, many alleles present in the population at the same time, and they compete with each other. And that has led to pretty good understanding of the rate of adaptation in these rapidly mutating populations, and how this rate depends, for example, on mutation rate, on the population size, etc., etc. But I started out by showing you this genealogical tree, and there is another very useful way of thinking about evolution, not as a branching process going forward, but as a coalescent process going backward. Now, suppose you're looking at the population at a given time, and this is a sexual population, so every individual has a single parent, right? So going back in time, everyone has a parent, but then some individuals are actually siblings, and they share a parent, so the number of ancestors always decreases. And therefore, in rather short time, this population, when tracked back, comes to a most recent common ancestor. And again, this is a stochastic process, but we understand everything about it, especially in the neutral case where all individuals are equally fit. And so this is called the Kingman coalescent. And in fact, all of our interpretation of genomic data is based on this understanding of kinship in the population. So the only thing is that statistical properties of this genealogy or coalescent tree depend very strongly on that assumption of neutrality, that all individuals were equivalent. So for example, if you simulate the population going forward and you say that they're all equivalent and therefore on average have the same number of offspring, and then you reconstruct the tree, this is the tree, this is a genealogy that you will get. It will look like this. Whereas if you do this in a population where individuals actually have fitness diversity, right? Some are more fit than other. So we have some distribution of fitness in the population. And then you simulate this, reconstruct the genealogy, look at the tree. You know, it doesn't take a lot of statistical analysis to realize that this tree is very different from that. But in fact, we also do understand these differences pretty well on statistical level. So this Kingman coalescent describing the neutral process is something that have been understood, what I guess 40 years ago, whereas a statistical understanding of the structure of genealogies in so fitness diverse populations is very recent. And in fact, Bernard Derrida was the first to point out that the statistics of these trees is actually very closely related to the statistics of trees describing the relatedness of spin glass ground states. So it makes a big difference how much selection, how much fitness diversity there is in the population. But the big question is, what sort of mutations contribute to evolution? And there are two different, very distinct possibilities. One possibility is that it's the rare strong mutations that occur, let's say, once every 2 years that drive antigenic change and render so a new emergent strain of virus insensitive to the antibodies that the human population has developed in response to the previous strain, right? So this rare strong mutation picture basically means that most of the time there is no fitness diversity in the population. Every so often a big mutation comes about. So the new clonal population arises and then grows up and makes another clonal population of identical, sort of new identical viruses. Although I should say that they're identical phenotypically, but not genetically. There are many other mutations that come in, they just don't have the effect on the fitness, they're effectively neutral. And then you expect that if you reconstruct the genealogy, in this case, it will be of this neutral coalescent type that I was just describing. So most of the time when you look at the population, all individuals are fitness equivalent, and these big mutations that change fitness come in very rare. You hardly ever see them in motion, right? And that's the picture that is pretty much dominating the thinking about viral evolution, flu in particular. But the alternative scenario is that actually very many weak mutations, small mutations, and they accumulate, and then the population is diverse in fitness, and then the fitter edge of the distribution is being amplified, and the less fit is going down, and that makes the whole fitness distribution in the population slide forward. So you have this continuous traveling wave of adaptation. So these are the two pictures. And as far as our biology knowledge is concerned, there is a strong discovery bias towards large effect mutations. If you're trying to study the effect of genetic substitutions, you're much more likely to identify a mutation with a large effect, right? So in particular, I was showing in the beginning this coloring of evolutionary tree into these subtypes, and then associating different colors here with particular substitutions in the active, or rather in the part of the protein recognized by the antibody. So by comparison, small effect mutations are hard to identify and difficult to quantify, but on the other hand, if you have very many of them, they can add up. So you have the situation that there is something there that you don't really see, but it has potentially a very large effect. So from a point of view of molecular genetics, this is really a nightmare, right? Dealing with these small effect mutations, you will never really be able to fully understand any one of them because it has a barely measurable and perhaps not even a measurable effect on its own. But to statistical physicists, this presents a rather obvious opportunity. So now you can try to describe the collective behavior of these swarms of small effect mutations without knowing each and every one by name, and without really getting very heavily into molecular biology. Well, fine, so let's try to do that. Let's assume the scenario that evolution of influenza is driven by lots of small effect mutations that accumulate. And we're going to build on this recent progress that I mentioned in the beginning, the subject, and see how far that can take us. So let's look again at this genealogical tree reconstructed from a sample of genomes. So if you're given a bunch of genomes, you can rather reliably reconstruct the genealogical tree from them, right? But we want to go beyond this genealogical relation. We want to be able to say something about their relative fitness. And we have assumed that there are many mutations that have happened here, and we're dealing with a broad distribution of fitness. So suppose we want to say something about fitness difference between any pair in this population. So what are we going to do? We're going to say, well, there were lots of mutations that fell in here on the tree. Some of them contributed, maybe blue are the ones that were contributing positively, and red ones were contributing negatively. And we basically have to take an incoherent sum over these things along the path, this genealogical path along the tree that connects our two leaves of interest. And what you expect is so the characteristic size of these fitness differentials you're going to find is going to be effectively diffusive. So it's going to be linear in scale linearly with genealogical distance. Well, that's kind of obvious, but the tricky part here is that Of course, fitness is not a passive traveler along these branches. It actually affects the branching process itself. And well, it's kind of obvious because remember, fitness virtually by definition is the number of surviving offspring. So if we look just at the bottom of the tree, we have nothing to say about the fitness because we don't know how many offsprings they're going to have. But if we look somewhere in the middle of the tree, you will find that some ancestors actually show up having just a few offspring, whereas some other ancestors have, sit on top of heavy clades and have a lot of offspring. So it's quite intuitive. You expect that these guys here are more fit than that, and you can actually calculate exactly how much this fact, the observation of sort of a large clade hanging off a given node, how much that observation biases your estimate of fitness of this ancestor relative to fitness distribution of population in the whole. Basically pushes it kind of with an exponential weighting factor, pushes it towards the nose of the distribution. So anyway, that's a very simple intuition, and you can turn this into a calculation of a joint probability distribution of fitness along the tree. So all you need to do for that is calculate how fitness propagates, remember, essentially diffusively along these branches, and then you just multiply these propagators along the tree, and you construct a joint probability distribution of fitness for all the nodes, conditional on the observation of a given tree. And well, that's kind of a testable prediction, and the first thing you want to do is test it in silico. You just want to take your favorite model, run it forward, take a sample, reconstruct the tree, construct this probability distribution, predict fitness. You want to see what it's going to be. Well, you know what? I'm not going to walk you through this slide. The answer is that it works reasonably well. But then you say, well, so what? This is just a model, and we've made all these assumptions, and our model already obeys these assumptions, and if we made wrong assumptions, it's completely irrelevant to anything. So the real test is to take the simple model into the wilderness of the real stuff. So let's go back to that seasonal influenza tree, and just focus on so the samples, so genomic sample that are present in any given year, and then ask yourself, can we predict the future, right? So of course, if you look historically at this tree, well, it's pretty clear what, how to predict, right? You just have to identify what's on this backbone. It's this backbone that's going forward. Those are the progenitors of the future. So all we have to do is look at these things and figure out which one of them should be on the backbone. Except it's not exactly obvious how to do it if, without the benefit of a hindsight. Well, on the other hand, if you can infer fitness and you believe your inference, then you can convince yourself how to convert. You can use that inference algorithm to try to identify most likely place in the tree where this progenitor of the future is going to be found. And so we do exactly that. We pull off sort of a sample of the genomes, we reconstruct the tree, we'll look at, do a little calculation on the structure of branching here, and remember, we're effectively looking for a clade here which shows a lot of branching happening in a short span of time. A lot of pattern of recent branching. And then we color code it, so I guess this red, or I guess it turns to brown, is so the highest probability of being a progenitor, and then right, this is done on historical data, so we do have the benefit of hindsight. You just attach the tree from the next year, and you see where it joined, and you see how well it predicts. Well, of course, this is because I picked a really good example. But we do have 15 years worth of data on which you can test, and this slide sort of summarizes what you find. So we can, we're looking now at the genetic distance between sort of the actual progenitor and predicted progenitor, and compare it to random pick, right? So that's our baseline, and then of course, with the benefit of a hindsight, there is always a best possible pick. So that's this line, and what you see is about a third of the time we pick the algorithm actually picks the best possible thing, and on average it does better than on average it does better than random. Well, you also see that you predict, but there's much room for improvement. But on the other hand, it would take me probably an hour to give you all 1,001 reasons for why this should have never worked in the first place. So the fact that this very simple model, basically with just one parameter that can be fixed very easily, achieves this level of prediction is not so shabby. But anyway, this was a rather dry summary slide, and I want to finish with something a little bit more fun. So my collaborator Richard Nair and his friend actually took this project all online. So as you see, here I'm presenting again this genealogical tree constructed from the data of yesterday, and it's only because I had to submit, send in the talk, last night that the data is not fresher than that. So this has gone live. It's available online. And what you see is the latest prediction. So there is this clade out there going forward, and we think that it's going to take over. And we are in a little disagreement with the World Health Organization, which chose this clade to build the flu vaccine of. And well, we don't know in this horse race which one is going to win, but we'll find out next year. And with this, I will conclude. So, so evolution is driven clearly not only by accumulation of this small mutations, as I just argued. Clearly, we know from molecular genetics that there are big mutations, and they have a big effect by definition. And what we're seeing is the result of both processes. So it's really a surprise that just by focusing on this dark matter, the accumulation of small effects mutations, one can actually achieve some level of short-term predictions simply building on statistical physics ideas. So the challenge is now to combine our simple treatment of this part of evolutionary story, that's this emergent simplicity part of the story, with the very real complexity part of the story, with understanding and identifying and learning how to combine our knowledge, so biology knowledge, which teaches us about these rare big mutation effects, with statistical physics and that's what we need to do better, and we need to do better. And I'll conclude with acknowledging my wonderful collaborators. We started this work with one of the postdocs at KITP, Adol Dayari, and decided that this wasn't enough data for him, so he moved to Facebook. But 90% of this work was done in collaboration with Richard Nair, also a former postdoc at KITP, who is now on his own at MPI, Max Planck in Tübingen. And Colin Russell is our other collaborator and flu expert, so we weren't entirely without any knowledge of biology. So thank you very much for listening. Yeah. Can you please speak up? Yeah.
Next year.
Well, we can do a little test here. We can ask how many people in this room actually got the flu vaccine, and I don't see too many hands up. So I don't think the penetrance of vaccine is at this stage close enough to be serious feedback. Right? We would like it to be, but. Also, if I had a little bit more time, I would have told you that this last year, and this is in defense of WHO, is a very complicated year with 3 new clades coming out, and some of you may already have partaken of that rather nasty strain that's out there. It's a fast-moving year.
Any other question?
Your evolutionary tree was sort of on a evolutionary axis, and I'm curious to know how that maps onto a biological axis of some phenotype, whether it is resistance to certain classes of antibodies or structure. Because it looked like it was evolving very linearly, but presumably it's doing a much more interesting multidimensional random walk in phenotypic space.
Well, it's basically trying to get away from where it was, right? Because, and that's, if you like, immunological landscape that it's evolving in, right? It has to innovate to get away from the immunity that was left behind by its ancestors, right? And it's a very interesting question of why is there only one branch going forward? Why don't we see deep splits coming in, right? Well, that's a very good question, right? But as far as, so dimensionality, maybe the best way of thinking about it is, well, it is a very high dimensional space, right? On the level of, viral epitopes, right? it can move in very many ways, right? But what seems to be happening is that the fastest moving thing dominates, right? And there are so many ways of going forward, right, that the tree never really comes back, right? But there's a non-trivial question of why is it going, why is it just taking one path in this multidimensional space as opposed to branching and splitting, and some other viruses do exactly that.
Boris, so I was just, taking off on that point that you raised. So is the constraint coming from some mechanism of replication of the virus? Is it coming from something that the virus sees in its interaction with the outside? Because if you take another virus like HIV or something else, the possibility of having these multiple splittings is more likely. So I'm just wondering whether the constraints are coming from the mechanism of the virus replication.
Well, I think the simplest way of understanding it is in terms of, if you like, clonal competition, the range of inhibition, right? So the question is, when you have a strain which is rising in dominance, right? How far out does it inhibit other strains, right? And as long as the probability of jumping out of that zone of suppression is small, right? That winner is going to be taking all, right? So I think that's sort of a relevant parameter. The range of cross-reactivity in and if you like, the speed of evolution. So how fast you can redesign the surface, also relevant bit of a protein surface to become unrecognizable to existing antibodies. We're actually thinking about that very question. It's I don't really know the answer.
Yeah, what are the other areas where you have enough data to apply this kind of modeling? I mean, for instance, mutations, caused due to environment, hereditary mutation, genetics, so on.
Well, it's a difficult general question that you're asking. One obvious, very interesting area is the immune system. So there is a major, sequencing effort going on antibody repertoire and T cell repertoire, and of course the immune system is an evolving system, and well, that's a very natural thing to try to look at, but there are surely others.
So perhaps we should take other questions offline and thank the speaker once more.